Our final speaker is Professor Marina Lynch, and she is a professor of cellular neuroscience at Trinity College Dublin. She's an adjunct professor of physiology at Trinity College Dublin, a fellow emeritus of the college, and a researcher in the Trinity College Institute of Neurosciences. She received a PhD from Trinity College and received post-doctoral training in King's College London and the National Institute for Medical Research at Mill Hill in London before returning to an academic position in Trinity College. Her research focuses on understanding the role of neuroinflammation in age-related and disease-related loss of neuronal function, in particular Alzheimer's disease. So just before Professor Lynch begins her talk, we're just going to pose to the audience a quick question as to, do they think there are likely to be different medications for males and females when it comes to these types of diseases? So the link should go into the chat now. Let's see. Have a look at the results and see what people think. Share the screen. So at the moment, 76% of people think yes, and 24% of people think no. So we'll let Professor Lynch take the floor and she can give us her talk.
Okay, I'll try to share. So can I just check that that's up now?
That's perfect. Thanks very much.
Okay. So as you can see, I have a fairly simple mind. I'm going to try and persuade you that there are 5 things that you should know about inflammation in the context of Alzheimer's disease. So I'm going to start straight away by telling you that it's estimated there are about 1 trillion cells in the brain, and about 100 billion of these are nerve cells. So nerve cells, of course, are hugely important because they are responsible for absolutely everything that we do, that we think, that we're capable of doing. And so the action all takes place here at the synapse. So the connection between one nerve cell and another is crucial to maintain the contact between cells and consequently ensure function. And it's suggested that there are about 1,000 trillion connections in the brain. Now, because neurons are so important, it's really key that there are other systems in the brain set up to try and keep them in good shape. And there are 2 cells in particular, the astrocytes, but especially the microglia, that have a particular role in protecting neurons. And I'm going to talk today about the microglial cells. So what do microglial cells do? Well, they're a little bit like the macrophages that Rukmani told you about. They're phagocytes. That means that they're responsible for engulfing anything bad that happens to occur in the brain. So their main job is to survey the environment, they sniff around the cells to check whether there's any evidence of danger to neurons or damage to any of the cells in the brain. And when they sense any danger or damage, they change their shape from this, what's called a ramified shape, to a much more amoeboid shape, indicating that they're now preparing to engulf the problem. And so they eliminate the difficulty. So this, I think, should, yes, here we are. I'm going to go back to the beginning again now, because I want to show you there's a blood vessel here. Here we have some microglia, and there, that was a laser beam causing damage to the blood vessel. And what you can see is that microglia cluster around this area of damage. And as you can see, there are little bits of yellow appearing. This is indicating that the microglia are actually engulfing the damage, and they change color to show this yellow damage that has been engulfed. Now, meantime, a little bit like what Manny said, these cells are able to repair. And so what they're doing is trying to repair the blood vessel. And you can see a little hint that the blood flow is beginning to go again through the blood vessel on account of the role that microglia have played here in repairing the damage, but also in engulfing those bits of tissue that were created as a result of that breach of the blood vessel. So bottom line here, microglia are phagocytes, and part of the role of the phagocytic role is in protection. Sorry, I need to go to the next slide. So one of the things that we know about Alzheimer's disease now is that microglial activation or neuroinflammation is one of the key indicators of the disease. So if you look at this MRI scan, a PET scan that was taken from a control patient control individual compared with an Alzheimer patient, what you can see is there's a very marked difference between them. And the color difference here, the green and the red, bright colors, are indicative of activated microglial cells because they pick up a particular dye that unactivated cells do not. But in addition to microglia activation and neuroinflammation, we also know that we have plaques and tangles in the brains of people with Alzheimer's disease and marked tissue loss. And you can see that there are very significant evidence of tissue loss here in this brain, which is an Alzheimer brain, compared with a controlled brain. Okay, so microglia, as I said, are the phagocytes. And so what they do is they hoover up any damage that takes place to cells, but also they're in the context of Alzheimer's disease, they're able to hoover up amyloid beta. Now, amyloid beta is what makes up those plaques that I mentioned earlier. And every body's brain produces amyloid. In fact, they're produced by neurons, but in very small quantities. And so there's no problem then for the microglia to hoover up this small quantity of A beta. But when this gets to be a much larger quantity, as we're moving into the pathological situation, it turns out that the microglia are no longer able to hoover it up. The cells are exhausted, and we're trying to figure out what the reason for this is. So to investigate this, we wanted to try and investigate whether or not there was a change in the metabolism of these microglia in a situation that might be akin to Alzheimer's disease. So very briefly, every cell makes their own energy by taking glucose from the blood and producing ATP, which is the way the cells produce energy. So there are two steps essentially in breaking down glucose. The first step is a process called glycolysis, and we get 2 ATPs as a result of this. The second part of the process is a much more efficient way of producing ATP. As you can see here, you actually end up getting somewhere over 30 ATP molecules when you completely break down glucose into carbon dioxide and water. Now, this first bit is called glycolysis. And as I said, it's a very inefficient way of producing energy. So the first thing we wanted to do was we wanted to ask the question, is it the case that when microglial cells are stimulated with something that causes serious inflammation, first, is there a change in the metabolism? And second, if there is, does this alter function? So what we did was we got microglial cells and we stimulated them with this stimulus that we know is capable of producing a huge amount of inflammation, and that's just measured in this instance by a molecule called TNF. So then we asked, is it possible that these cells now change their metabolism? And without going into any detail, all I'd like you to do is to notice that these dark red dots, which are the stimulated microglial data, compared with the blue, have a higher value here. This is indicating to us that they're using glycolysis. They've switched from another way of producing energy to glycolysis, the inefficient way. And when we investigated 2 measures of function, that is the ability of the cells to move, for instance, to move towards damage, this was reduced, and the ability of the cells to engulf amyloid was also reduced with this stimulus. So we have cells that are inflammatory, they're glycolytic, and dysfunctional. So one of the things that's been known for quite some time is that there are a number of diseases that are different in males and females. For example, right at the end of the spectrum on the right-hand side of this slide is autism, and we know that about 4 times more boys than girls suffer from autism. And on the left-hand side of the slide, we see that Alzheimer's disease is predominantly in females, with several other diseases that affect the CNS somewhere in between, but also indicating sex-related differences. With regard to Alzheimer's disease, we know that between 30 and 35% of all the patients in the world right now are males, and up to 70% are females. So the bias is very much towards females getting Alzheimer's disease compared with males. So we used a model of Alzheimer's disease. It's a mouse model of Alzheimer's disease. And the first thing we wanted to do was to find out, is there any difference in the amount of amyloid plaques that are in the female compared to the male brain? And sure enough, we see that there's a big difference between the females and the males, with more amyloid in the females compared to the males. And here, in a very early stage, you can see many more red dots, which is indicative of this in the female brain compared to the male. And at the same time, when you measure the area covered by these plaques, you see that there's an increase in females compared to males as well. Now, I don't want to make a huge point about this, but we wondered if these cells were inflammatory, the way I mentioned earlier about the inflammation induced by that stimulus. And all I want to do is ask you to appreciate that this panel on the right-hand side of the slide, which indicates the Alzheimer females, have many, many more red genes than any of the other panels. This is indicating to us that inflammation is much greater in these mice, the Alzheimer mouse, females compared with males, and compared with mice that are not these models, they're control mice. And when we took the cells from these and examined, again, the metabolism, again, I just want you to focus on this dark red or brown picture here. You see that there are these cells from the female APP mice, or the Alzheimer model mouse, have a much greater glycolysis. This is indicating to us that they've switched their metabolism to this inefficient way of producing ATP. And at the same time, as you can see here, the amount of lactate in the brain is increased in these mice. And what we know about that, as everybody knows, is that lactate makes you tired. So we're thinking that this production of the lactate plus the inefficient way of producing ATP might well explain the exhaustion that these cells experience as we go from normal into an Alzheimer's state. Now, the other thing that we discovered about these cells is that they look completely different. So if you look at these, now these are computer-generated images from the real slides. What you can see is that we have a large number of these amoeboid-looking cells in the brains of the male, but not the brains of the female Alzheimer mice. And we did a huge amount of calculation on these to figure out whether or not they truly were different. And I can assure you that they are. The size of the cell, the length of the processes, all sorts of different things are different in the females compared with the males. Now, you might ask, what's the significance of that? Well, I mentioned earlier that these cells that look more rounded and amoeboid are more phagocytic compared with these guys here. And we did several studies to try and investigate whether this was true or not. And these are results from one such study where we had cells that were either that they had with processes, cells with processes, the ramified versions, or the cells that were very rounded, amoeboid. And we asked whether these cells took up a different amount of amyloid. And the amyloid is shown in green here. And you can see that the cells with processes do take up a little bit of amyloid, but not nearly so much as the cells that are rounded, in other words, amoeboid cells. So we now know that the cells in the brains of the male Alzheimer mouse take up more amyloid. So we wondered, might this be the case also in patients? So we got tissue from the Netherlands Brain Bank from male and female individuals. Of course, this is post-mortem tissue, and we